everyone. Happy New Year. Great to see everyone again for another semester of the HPL seminar series. Today we'll be hearing from Dr. Jillian Beveridge. Dr. Beveridge is assistant staff in the Biomedical Engineering Department at the Cleveland Clinic and an assistant professor at Case Western Reserve University, where her research investigates the mechanisms that drive post-traumatic osteoarthritis after intra-articular joint injury. Jillian did her undergraduate studies in sport rehabilitation at Acadia University and received her bachelor's in 2002. Following, Jillian pursued a PhD here at the University of Calgary, working with Dr. Frank in biomedical engineering. She completed her PhD on mechanical mechanisms of cartilage damage in ovine models of joint injury in 2012. Dr. Beveridge then pursued two postdoctoral fellowships, the first at the University of Calgary with Dr. Janet Wronski, and the second at Brown University with Dr. Braden Fleming. Her research interests focus on knee joint injuries and the structural and functional changes that may contribute to early onset osteoarthritis. Her vision is to inform novel solutions to reduce the risk and severity of post-traumatic OA. Jillian's proudest accomplishment is contributing to the team effort of bringing the now FDA-approved Bridged Enhanced ACL repair, termed BEAR, a tissue-engineered scaffold that stimulates the native ACL to heal. She even performed a BEAR procedure on a pig. In the future, she is looking forward to completing the construction of the new biomechanics lab at the Cleveland Clinic. One project she is excited about is the collaborative work exploring neuromuscular function in ACL patients who've received the BEAR implants. As a fun fact, Jillian loves sports and is fiercely competitive. She has been a member of elite sailing, skiing, and triathlon teams. As a reminder, I'll first turn it over to Dr. Beveridge for her presentation, and then we will have a question and answer period to follow. And at that time, I'll remind everyone how to submit a question. So with that, Jillian, you're welcome to start sharing your screen and begin your presentation when you're ready.
Great. Well, thanks so much for that introduction. I think you've kind of hit all the highlights of my training and where I am today. And my goal is to actually share with you a lot of the work that I've done that spans from my time in Calgary doing my PhD up until kind of the present. And I'll touch on a few elements of where I'm headed next. And throughout all of this, my interest has been on ACL function and understanding how the ACL will modulate joint biomechanics and contribute to PTOA risk. So as Emily said, I did my PhD here in Calgary, and I was here for, I guess, just over 10 years. I first did my work with Cy Frank over in McKeg and was co-supervised by Nigel Shrive. And then after my PhD, I did a postdoc with Janet Wronski. And the picture I have of Calgary here is obviously dated, but it looked a lot like that when I first moved. So after I finished my PhD and that first postdoc, I had the opportunity to go to Brown University. I was there for 3.5 years, and I worked with Braden Fleming at the Rhode Island Hospital and Lifespan, which is kind of this building on the bottom right. And Brown was a really great experience. It was a beautiful camp, very New England, very picturesque, and I had like just an all-around fantastic experience. And then I've been at the Cleveland Clinic now for 3 years, which I can hardly believe myself. But once I was there, I established the Movement and Biomechanics Lab, and I'm in the Learner Research Institute, which is this building here on the left. And being the Cleveland Clinic, it really is much bigger than I ever imagined. It's a huge, huge organization. And kind of one of our flagship buildings is right here on the right. My perspective is, it's a hospital organization. And then when you go in, you kind of question whether in a hospital or in a hotel. Like it really is quite incredible. And there's about just short of 200 principal investigators in the Lerner Research Institute. So although it's a hospital system, they have a whole kind of institute that's totally dedicated to research. So that's where I am. So the overarching question through all of this that has guided my research is why and by which mechanisms do patients develop post-traumatic osteoarthritis after ACL injury? And how I've approached that question, when you look at the spectrum or the research continuum of the different scales you can work in, I've tended to gravitate towards working in large animal models. So looking at the joint as, or considering the joint as an organ. So you can look at the living microenvironment, you can use traditionally more invasive methods. And then I've also done some work more at the human level. So the joint series, looking at, well, how do each of the joints relate to each other? How does the human body move? How do all of that gets coordinated? The beauty of where I am is that I feel like I can scale down towards to look at more tissue, the function of tissue. So that was a lot of the work at Brown. So you could use tissue explants, you could look at structure and composition, and you can actually build some computational models. On the other hand, I'm also poised to scale up. I've got some great clinical collaborators, especially where I am. And so you could actually scale up some of the techniques and approaches that you're working at the joint series up to the whole being. You could have large scale randomized control trials, and you could look at patient reported outcomes and these large multi-center studies. So hopefully, we're in kinesiology and sport medicine. So I think we probably all know our anatomy, but the ACL links the femur to the tibia. It provides joint stability primarily in anterior tibial translation and internal, it resists internal tibial rotation. And anterior cruciate ligament tear is a concern at least in the United States and in Canada as well, with up to 250,000 tears reported annually. This corresponds to approximately 1 in 3,000 tears per person, and about 1.4 million ACL non-contact injuries globally. One of the big question marks after ACL injury is, we know that it leads to post-traumatic osteoarthritis, which is a degenerative condition in the knee or other joints that's characterized by loss of cartilage, bony changes, pain, and joint dysfunction. This is an older graph, but I really like it because it explains how the term came to be the young patient with the old knee. And so on the x-axis is the age of the population that develop osteoarthritis. So men are in the squares, women are in the circles. And you can see that as you age, a larger proportion of the population, up around 50%, by the time they're at least 65, will have OA. Whereas there's these outliers here on the left. And this is the proportion of people who sustain either an ACL injury or a meniscus injury. And you can see that up to 50 or 60, or nearly 60% of those people, they're developing OA when they're 35, which is a huge problem when you consider that there's no disease modifying therapies, including ACL reconstruction. So understanding the pathway from ACL injury to PTOA is really an important clinical problem. From a mechanical standpoint, which is the tack that I've kind of looked at it, we know that there's changes in compressive stress. We know there's changes in shear stress between the articulating surfaces. We think there's increased friction, which in turn lead to greater tissue strain and cell deformation. So when you look at a number of different factors that modulate this relationship, I've really honed in on the kinematics or the movement. I've looked at the soft tissue biomechanical properties of the joints or of the tissues that constrain joint motion. And to a lesser extent, I've kind of dabbled a little bit in kinetic loads and starting to dabble a little bit more in the biology. So those are kind of approaching the problem from a mechanics point of view. So there are 4 main research questions that I'll share some of my work with you guys today. So after ACL injury, first, what are the mechanical mechanisms contributing to post-traumatic OA? Second, can changes in ACL function be quantified non-invasively? Third, Does ACL graft function modulate contact mechanics? And lastly, how much does the mechanobiology change? And is there a point of no return on this pathway to PTOA? So the first one, mechanical mechanisms and PTOA. This was the work that I did at the University of Calgary in my PhD. And for some of you, this actually may look familiar. So what we did was we used a large animal model of ACL injury, and we attached these rigid marker sets directly to the bone using this removable system. And because the relationship of the markers to the bone doesn't change, we can back out the tibiofemoral kinematics. So this is one of our animals walking on the treadmill. You can see the mocap, the reflective markers here on the tibia and here on the femur. And so we'd register those positions to the bones and we'd back out our kinematics. This is an example of the relative proximity between the bottom, the tibial plateaus here on the bottom, the femoral condyles, from an in vivo gait cycle. Red is closer together, blue is further apart. And this, a full gait cycle is kind of hoof strike to hoof strike. And so we'd use those contact mechanics to back out different parameters. And at the same time, in a two-dimensional way, we could actually draw out where we saw cartilage damage, which is represented in blue. And then we overlaid where we saw cartilage damage and how severe that was for each animal. So we could get a sense of where the damage happened and how bad was the damage. And what we saw is one of the mechanical mechanisms that we looked at was relative sliding velocity. So how quickly does the tibia slide relative to the femur? And what we saw was that the animals with worse cartilage damage here on the y-axis had a greater increase in relative velocity after injury. And so there was a small sample size, but I would argue that the relationship is really quite compelling. The other piece to this was that although there's a relationship, which means that there's a variation in the response to ACL injury. And this is really striking. Again, something striking to me is that here's the example. Here are two animals. There's one animal here on the left, one animal here on the right. The femur's on the top, tibia's on the bottom. And this is the change in the proximity or the change in relative distance between the two articulating surfaces after injury. So here on the left, you can see there's a lot of greens and blues, which corresponds to roughly no change in the distance. So their mechanics were really stable before and after injury. And we really saw no gross cartilage damage. Conversely, you look at the animal on the right, and you see these really dramatic differences. So regions of the tibia here are now 8 millimeters closer together after injury and up to 8 millimeters further apart. So you have this huge shift, which we showed was related to this measure of congruence, which again was related to the amount of cartilage damage we saw. So even though it's the same breed of animal, the same sex, the same surgery, the same amount of activity, all of the variables you could control, there's these really dramatic changes in mechanics. So that kind of wrapped up my PhD. And what I really argued was that there's strong evidence to show that when the mechanics are abnormal, you'll get increased cartilage damage when there's no ACL. We did an ACL transection model. So kind of the next logical step was to test whether we saw the same mechanical mechanisms in the sheep, whether those were similar in human. And so I started my first postdoctoral fellowship, and that's where I worked with Janet Ronsky just as the biplane video radiography system was coming online. And so I did just as I wanted to do. I wanted to test whether I saw similar contact paths in humans and sheep. So on the left, here's a little schematic of a biplane system. So the idea is that you've got two X-ray sources, and then you have these image detectors here in the back, and where the two X-ray beams cross, you have your subject move through that field of view, and you can construct the 3D kinematics by using the underlying X-ray images. You have a subject-specific bone model that you can overlay in this virtual 3D calibrated space until the edges of the bone model match the underlying X-rays for both views. I'm just showing one. And then you get your 3D orientation. And so from that work, we found it was actually really difficult to find subjects that were still ACL deficient and weren't reconstructed. So here's an example where the blue paths are the paths of the ACL deficient, either time point or the ACL deficient knee in the human. And the red is the intact time point or the one subject's contralateral limb paths. And you can see in the sheep, you see this on the femur, this anterior and medial translation of the contact path here in blue. And you sort of see the same thing on the human. And you can see the same thing happening on the tibia, these contact paths. They shift kind of in the same direction on the medial condyle. The lateral condyle is much less obvious in the human. So that could be different subject-specific difference, or it could be differences between the animal model and the human kinematics. But there was just some preliminary evidence to suggest that these mechanisms are probably shared between the two, the animal model and what actually happens clinically. So speaking of clinically, we did a lot of this work in ACL deficiency, but really what happens clinically is that you get your ACL reconstructed. So the reconstruction consists of a tendon that is strung through these bone tunnels to replace where the ACL used to be. And there's about 100,000 ACL reconstructions performed in the US annually, and that number continues to increase. And it was increasing right up until COVID, when all the kids stopped playing sport. What we do know about ACL reconstruction, like I said earlier, it doesn't prevent PTOA, and we know that it doesn't restore 3D joint motion completely. So here's some data, some biplane video radiography data that I didn't do, but this came from Scott Tashman's lab, where subjects jumped up and down in the field of view on one leg. And you can see that the ACL subjects, their reconstructed knee were more externally rotated compared to their contralateral. And the tibia was more anteriorly displaced, not by a lot, but there's enough that they are different. And so you ask the question, why? Well, you've done a surgery that provides some gross joint stability, but it still doesn't recapitulate the native kinematics. So why? If you look here on the left, on the top, this is the native ACL, and on the bottom is an ACL graft. So you can see just visually, the morphology is different, that the graft doesn't recapitulate the native vascularization or innervation, and they actually have the tissues have different structural properties. Other confounding factors are that there's injuries at the time of concomitant injuries, so injuries to the cartilage surfaces or menisci when the ACL is injured. And also an important factor is that the ACL surgery itself is probably a second inflammatory insult to the joint. So all of those reasons could contribute to why we don't see the 3D biomechanics restored completely. This would kind of really, we went back to the human to see if there are shared mechanical mechanisms, and we gravitated actually back to the animal model. And this is some more work that I'd done in Cy Frank's group. And so we acknowledge all of these differences in the ACL graft and asked the question, well, what if we did the perfect ACL surgery? So what if we used the native ACL as the graft? And we did that in the sheep model by actually taking a reamer, coring a reamer, and we cored from the outside in around the footprint of the femoral ACL origin. And then once we cored around that origin, the whole ACL was kind of attached to this bone block. We fixed the bone block exactly where it was beforehand. And so in this way, we replicated the drilling aspects that occur with the traditional ACL reconstruction, but then we just used the native graft, so we were able to use the same 3D morphology and mechanical properties of the tissue. So we, like before we did with our ACL deficient animals, we measured the kinematics before the surgery, and then we measured them again afterwards. And what we saw, to our surprise, was that following ACL, this idealized ACL reconstruction, or IACLR, for some animals, by 20 weeks after surgery, their kinematics were different from a sham procedure, and their kinematics were changing in a direction that you would expect with ACL deficiency or an impaired ACL. So that we saw greater tibial internal rotation, and we saw greater anterior tibial displacement at mid-stance. So when we looked at those results, we kind of questioned, we said, well, if we've done the best ACL surgery we could possibly do, what else had to be changing to drive these changing kinematics? And so we kind of landed that the biology must be changing, and maybe it's the structural properties of the graft that is changing. And of course, it's impossible to measure the structural properties of an intraarticular tissue in vivo, except there was an opportunity for me to go to Brown, and this is where I started my second postdoc. So Braden Fleming was working on these methods to predict ACL functional properties or structural properties from magnetic resonance imaging. So the pipeline, or what we would do, is that we would take an MRI of the ACL. This is the ACL in blue, and you would segment it out from the image, and that's blue. So for every slice of the image, you say this is the ACL, and then for every voxel, which is a 3-dimensional pixel, we could measure the signal relaxation of the MRI. So this is a specialized, excuse me, a specialized sequence, excuse me, called T2 star. And so the T2 star sequence, you take multiple snapshots of these voxels as their signal decays over time. And what we know about T2 star is that the rate of signal decay is related to the collagen organization and the water content of that tissue. So from the segmentation, we could extract a metric of quantity, and from the relaxation time, we could get a measure of quality. And so the relaxation times in the ACL range from 0 to about 50 milliseconds. And with the shorter T2 relaxation times, those are indicative of more organized collagen and less water content. So in this case, if you look at the next graphs, blue is good and yellow is bad. So what we saw in building these prediction models is that the distribution of these T2 star voxels changes with ACL healing. So at 6 weeks after ACL surgery, you can see this histogram where the distribution of the number of voxels that have a short T2 star relaxation time in blue, the volume is about the same, or maybe a little bit more than those with yellow. But by 24 weeks after ACL surgery, you can see a dramatic change in distribution. So we leveraged this information to build a statistical model that would predict the healing ACL stiffness, yield load, and failure load. So we had the model predicted stiffness, and we could validate that with the actual stiffness that we measured after the surgery. So this was actually a pig model of ACL surgery. So we could do the surgery, we could do the MRIs, and then at the end of the study, we'd euthanize and do the direct measures as a gold standard. The other piece, Braden Fleming's group was really interested in understanding ACL healing and ACL function, and I was too, but I still gravitated back to, well, what is the relationship between ACL stiffness and cartilage damage, thinking in my mind back to the results of that ICLR study. So from the same animals that we built these statistical models, we had our direct measures of ACL stiffness, and you can see that with healing time with 6, 12, and 24 weeks, stiffness increases post-operatively, which is what you'd hope to see. And then we took those 24-week stiffness values and correlated them to the gross cartilage damage that we saw at 24 weeks. We did that for each of the time points. And what we saw was that only at 24 weeks did a relationship emerge between stiffness and cartilage score. So animals that had stiffer healing ACLs had less cartilage damage than animals with more compliant ACLs. And here's an example of what it looks like on MRI. So the top animal, these are 2 animals, same procedure. Again, we're seeing the variation where the predicted stiffness, you can see a lot of yellow, was about, they're just under 40 Newton millimeters. In the 24-week one, the other animal had 122 Newton meters. And when you predicted the stiffness for these two animal models, they again did really well. So we predicted these values and they lined up really well with the cartilage scores. So the take home from this was that it appears that ACL stiffness is also related to the cartilage score. So on the one hand, I've got data from the sheet model that shows that the joint kinematics are related to cartilage damage, the abnormal mechanics. And at the same time, I've got a measure of ACL stiffness or predicted stiffness that also relates to cartilage score. So if I could combine the two, then I could understand what happens, do abnormal mechanics change, and does ACL stiffness modulate those, and in turn lead to more cartilage damage if the mechanics are abnormal after ACLR. So this led to another study where I was building on trying to kind of combine everything together. And I took a model from another prediction model that was based on just standard MRI imaging. So this was not the signal decay sequence. This was just a model that you get, so you could extract, again, the signal intensity, where darker signal on MRI means more organized tissue. And you could segment out the volume of the graft. And when you put these two together and predict the 5-year HOP score, so HOP score for in the clinic, if you have a patient who had an ACL reconstruction, one of the functional tests that you get them to do to gauge return to sport readiness is to do a one-leg hop for distance. So they take off and land on the same limb, and they express, and you look at if the limb is, if you reach about 90% symmetry to your uninjured limb, it's a good gauge of like you're functionally ready to return to sport or return to activities. So Braden Fleming had been following a cohort of about 60 ACL reconstruction subjects out to 5 years. So this is their 5-year HOP score. We looked at their predicted ACL, we use signal intensity and volume to predict that HOP score. And below, behold, we saw a relationship. So this MRI-based metric of graft function again relates to a global joint function, a global function. So I kind of wanted to take that idea and drill down a little bit further. And that led to this grant submission in the United States called the K99R00. And it's a mechanism that supports training in your transition to an independent investigator role. So the K99 portion is a 2-year mentored study. And then the R00 gives you 3 years of funding, or 3 years of kind of startup funding, basically, to pursue your independent research. And it was, as a Canadian, it was the only grant I would qualify for. And it's very, very competitive. So I said, sure, I'll apply. And so we put it in, and actually on the second try, we got it. So I intended to go down to the United States for 2 years, and 6 years later, I'm still there. But anyway, so the graft function and contact mechanics, that's what I wanted to do. I wanted to pull all of these bits and pieces of work that I had done into a single study. And so I wanted to understand, if my predicted graft stiffness is low, do I get increased joint motion abnormality? So for the K99 portion, I did it at Brown with Dr. Fleming, and I leveraged his long-term ACL graft function and contact mechanics, or the clinical cohort, to extract those measures. So these are ACL reconstructed subjects who were between 10 and 15 years post-reconstruction, at a time where we would expect to see cartilage damage in a clinical population. And then I proposed for the R00 was that I would then use an animal model to define the threshold and time course of those changes. So it would be a longitudinal study. So from the K99, the mentored study, we used biplane video radiography to capture the joint kinematics during a 1-leg hop for landing. And what we saw was that in the ACL reconstructed subjects, their tibias were translated almost 5 millimeters more anteriorly than control subjects. And then we took that range and anterior tibial position at the time of contact and looked at how does that relate to the predicted ACL stiffness. And again, we saw a relationship, perhaps not a home run, but there is probably something there. So this is one subject in red. He has a low predicted graft stiffness and high anterior tibial position at contact versus this subject in purple, which had a much higher predicted stiffness, had one of the lower anterior tibial displacements. So there's some evidence that I think that graft function relates to the joint kinematics, but the plot twist is this. This is the contralateral. So blue dots are the ACLR and the white are control subjects. And the blue left and right are symmetrical, as are the controls. So whatever is happening in the ACL reconstructed subjects, it's happening in both limbs. And we're not sure whether this is just the way they are. So they actually reached, their kinematics returned to what they were before. Maybe that's why they got injured in the first place. Or whether this is some sort of systemic change that tries to match the contralateral to the surgical limb so that you reach symmetry. So this is kind of like a big Pandora's box that we have opened and we're trying to make sense of. And so we're starting to do some thoughts of how we could start to tease apart this information in a new study. So the cartilage thickness mapping from these subjects is ongoing. And I show this slide only for the purpose that if you look across the femurs and the tibias, these are a mix of ACL reconstructed, control, male and female. You can see that there's differences. So blue is thicker cartilage, and red is thinner cartilage. And what we're currently working on now is a way to take this visualization where I visually see that there's differences and come up with a quantitative metric that captures these differences. So it's an area of ongoing investigation, and others have taken, there's a number of different approaches out there, and we're looking to see what's going to suit our data and capture what we see. So that wraps up the measured study that I did at Brown. At that point, I was ready to start my position at Cleveland. And so I came out as soon as I started. We started with the study to build our animal model to achieve the R00 study objective, which was, if you remember, to define a threshold and time course of changes in the graft stiffness and joint motion abnormalities. And I wanted to use that IACLR model. So that way I could look at purely the inflammatory components and have the hypothesis that if I do the perfect reconstruction and the graft still changes over time, then it's the biology driving mechanics, which is kind of a more provocative hypothesis. So I had two main aims with that study. One was to take that MRT2 star-based prediction models that I had developed at Brown and make sure that they're tuned specifically to the IACLR model. Secondly, I wanted to determine whether IACLR impairs the predicted graft stiffness, and then to define that magnitude and time course of changes related to joint kinematics. So I could do a longitudinal study, not just the cross-section at the end. So we designed it so that we'd have 24 mature Yucatan mini pigs. Like all the previous animal studies, we'd record intact joint motion, we would do the surgery, and then at either 6, 12, or 24 weeks, we'd record those metrics again alongside the MRI, and we do biomechanical testing of the ACLs to tune the prediction models. Some of our key outcome measures that we're extracting are the 6 degree of freedom motion, as well as the contact mechanics, the ACL MR imaging, the gradient echo here, the GRE is the conventional sequence that we're using, But since starting at the Cleveland Clinic, I've had the opportunity to collaborate with Xiaozhuan Li, who is one of the preeminent musculoskeletal MR scientists, and she's developed some really great sequences that are both specific to cartilage changes as well as ligament changes. So we're testing her sequence as well. And then at the end, the graft, quote unquote graft, biomechanics. So we'll extract the ligament structural properties. So I'll show you a little bit, some preliminary data and kind of the work that we've done at Cleveland to get this far. So the premise was that with the pigs, I had considered doing biplane video radiography with them to extract the kinematics, but the reality is that their conformation of the shape of their body, their bellies are so big and dense that you can't actually image their femurs with X-ray. So you have to use the bone plate or some sort of bone plate system to extract the kinematics with sufficient accuracy. So knowing how well that worked in sheep, the idea was to take that method and apply it to the pigs. One of the challenges that, for the study is that unlike the sheep, we're planning to do MRI. And so you can see in the sheep, or if you recall, the marker sets and bone plates, they're made out of stainless steel, which is a big no-no for MR imaging. So the first step was to come up with or design a custom-based base plate that is both biocompatible and MR compatible. So we did that, and then we printed these 3D printed these marker sets that could be rigidly attached to the base plates. And then we wanted to use hardware that would give us a really good fixation and know that it's screwed into the same place every time, but that we could remove the metal pieces so that when we do MRI, there's no artifact. From a computational point of view, how we back out the kinematics is just the same concept as we did in sheep. One of the differences between our pipeline and the sheep pipeline was that when you go to define your anatomical coordinate systems of the bone, we did it manually in the sheep, and it required taking a handheld digitizer to digitize points on the bone that you picked. And of course, if I digitize points and someone else digitizes the same points, or what they think are the same points, those two things may not be the same. So we wanted a fully automated way to do that. And the first step was to figure out where these marker sets would be relative to the bone. So we thought, well, what if the base plate is on the bone? What if we put a marker registration or some sort of registration grid in the base plate? So that's what we did. That's this pink grid here. This is an MRI of the base plate. So we've made actually the material of the grid MR opaque. So we also custom designed bone screws, which was way more difficult than I thought. I guess I have an appreciation for these orthopedic companies who charge thousands of dollars for like bone screws. And now I appreciate the amount of work that goes into them. So anyway, everything's MR compatible. We've got this registration grid. We have a way of seeing where the grid is in the same space as the bone. And then so we can actually segment out that grid. We know where the grid is in our CAD model. And then we actually, once these marker sets are 3D printed, we will actually manually measure them with the digitizer. So we know that the 3D print, if there were any inaccuracies in that, we can account for it. And then we also machine these little registration divots. So we knew we had a way of figuring out where those marker sets were relative to this CAD registration grid. We knew where the registration grid was relative to the MRI. And the last piece was that we modified this automated algorithm that makes an anatomical coordinate system based on bone shape. So we kind of had this conceptual framework to do the registration. And then when you look at our cumulative error, it's about less than 0.2 of a millimeter to figure out where the spheres are in space. We know it's less than 1/10 of a millimeter to register the plate to the CAD model. And it's about 3/10 of a millimeter, or the size of one voxel, MRI voxel, to register the CAD model back to MRI. So and then of course we have the era of the motion capture cameras. So when you put that all together, we know our precision, we're about a millimeter or less. And this is all great in technical engineering, it's fantastic, but none of it matters if our subject doesn't walk on the treadmill. So here's one of our piggy wiggies. She's been trained to walk on the treadmill, and it's not perfectly in sync, but you can see just the mocap view of the marker sets as she walks. And then when you apply the pipeline, you can get the 3D motion of the tibia relative to the femur. So this was one of our girls, and this is 10 consecutive gait cycles for her. And we collect it's actually pretty quick. The hardest part is getting the marker sets onto the base plate. But we can collect about 200 strides or so while they walk. And then we wrap up the kinematics. So the other piece to this work was testing the T2 star sequences and translating, making sure that the outcomes that we got at Brown, we could reproduce in Cleveland. So in the top, this was an animal at Brown. So the animal's just anesthetized. So they're asleep. And then we put on the coil and we put them into the Met. And then we do the same thing here in Cleveland. And it's quite adventurous. In Cleveland, we had to trek these guys across campus and put them in a clinical space in the morning. All of the MRI techs were really eager when we had a piggy MRI day, because it was very unique and different. So here's an MRI. You can see the ACL segmented here. Again, we're using the same T2 star relaxation sequence that we used at Brown. You can actually see this little black spot here is the screw from our bone plate. So we looked at the T2 relaxation time, the R square of that fit, which is a measure of how well we've fitted our estimation of T2 star to the signal relaxation. So blue is a perfect fit, 1. And so we've got a lot of blue. We're really confident in our sequences. And then our 2 animals, when you look at left and right, they're similar even with the bone plate. So we know that adding the bone plate didn't affect T2 star. We've got really good R square fits, and we're reasonably close to the metrics that we achieved at Brown. The other thing that we did, we want to know, obviously we want to be able to try and do the IACLR surgery. So that was actually a big challenge in the pig, and frankly, it didn't work. So we tried it, and the shape of the pig joint is just different enough that we couldn't drill down to the femoral origin of the ACL without really damaging some of the lateral structures of the porcine lower limb. So we did not an about face, but we switched to a procedure that was again developed at U Calgary with Cy Frank, Nigel Shrive, and Dave Hart, where instead of doing the coring around all the way around the ACL, we actually just drilled from the inside out. We made 3 drill holes around the femoral origin. So we introduced that interarticular drilling, which we know is pro-inflammatory, but we were absolutely certain that we didn't damage the ACL. So we called this the IA drill model, or for interarticular drilling, and we compared that to just a sham. We also measured the hind limb loading in these animals over time, both with the plates as well. We had a separate group for the kinematics, just as pilot, and a separate group for our drill and sham animals. And so you can see that the hind limb loading in the sham is very reproducible from baseline to 20 weeks. And it becomes more variable in the interarticular drill animals. This is just an example of one animal. So we don't yet know why or whether that means anything, but we know we can measure it. And then we have an example of the same animal where we've mapped their T2 star at 6 weeks and 24 weeks after drilling. And we really actually didn't see a large change. So this is their left limb. The only thing that seemed to kind of the median T2 star was the same, the distributions were the same, the averages were the same. The only thing that showed any kind of sensitivity to the intra-articular drill procedure was the standard deviation. So it seems to be elevated at 6 and 24 weeks, and it seems that that spread in values seems to change a little bit with time. So we're still digging into what that means, but we're confident that we're picking up on subtle differences. So the last step would be to test these ligaments mechanically. So do an experimental tensile test to extract the structural properties. And so basically you extract them from the force displacement curve, and we were in the process of building a jig at Cleveland to do this. And we're ready to go, and we just took a moment and a step back because we looked at the bigger picture and said, we have an opportunity to do something really different. So Will Zahler's a postdoc, and we submitted, this would be the equivalent, the F32 would be an equivalent of like a CIHR fellowship, so a national level fellowship, where we propose is that in addition to getting kind of the basic stiffness, yield load, failure load of the tissue, we could actually build a finite element model that is informed based on the MR T2 star mapping. So we'd have the geometry, for example, from the ACL from the MR images, but then we could take the distribution of these T2 star voxels, map it to the geometry, and then we'd have a way of calibrating it because you could do some non-failure testing and digital image correlation so you get the surface strain. So you'd have a way of tuning the material properties that you assign to the model, your ACL model, and then you could actually validate it during destructive testing to say that are we modeling this correctly? And then we have two strengths where you could then drive your model by the in vivo kinematics that were recorded. And then we've got this whole data set from Brown where we've mapped changes in ACL T2 star properties as a function of healing. So you could actually start applying those ACL healing changes to the finite element model. So it would be a slick way to do a lot of simulation of how the ACL bears load in health and post-operatively. So that's currently in the works. So that's kind of the main study. That's what we're working towards. And really the elephant in the room is that I haven't acknowledged the changes in the biology. So in addition to the outcome measures that we proposed in the grant, we also did some synovial fluid arthrocentesis where we sucked out a bunch of synovial fluid. And then we're currently running some multiplex assays for cytokine concentration, as well as some of the pro-inflammatory MMPs. So that's actually happening this week. And so we're hoping, fingers crossed, to have some data by next week. So really kind of that wraps up my whole trajectory of where I've started with asking questions about the mechanical mechanisms of PTOA. And I think I've got some data to show that we think surface velocities, which are related to shear stress, and incongruity, which are probably related to compressive stress, that are important considerations in the mechanisms that damage cartilage after ACL injury. We've developed non-invasive measures of ACL function, so the T2 star sequence and the relaxation time that's related to composition and collagen organization, leveraging that to predict in vivo tissue changes is an exciting new area. I think that there's at least some data to suggest that ACL graft function in people and the contact mechanics are related. Although yet, like I said, we sort of opened Pandora's box, so we're still exploring that. And then the last question was, how much does the mechanobiology change, and is there a point of no return? And that really is to be determined as our results trickle in from the R00 study. So I think I do want to get to questions, but I've just got a couple more slides that I think are maybe relative to some of the work that's ongoing. And this is where I'm going next. So again, if you think about the research continuum with the mobile lab, I've been working in this realm, and one of the next steps is I really want to get up to here. So working with people again. So we're in the process of building the mobile lab. It's about 1,400 square feet. So it's nice and big. We've got a runway that's 25 feet long. We're building a biplane system there. So it's 14 by 14 foot overhead rails. So this is much bigger than the room and space that currently houses the biplane system over in the McKeag Institute. In addition to, I'll focus on the mocaps stuff. The other really neat thing about this system is that we're also installing these digital flat panels to capture the images. So what that does is that will give us almost like a super resolution and really clear images to do the 3D reconstructions. A 10 camera Vicon system is going in, and we're going to put in an instrumented treadmill, which is right here. And it's going to sit on this whole floor. One of the benefits, it's funny, Walter was asking me, well, who's at the Cleveland Clinic for biomechanics? And I said, just me. And it's kind of true. But I inherited this space that was really well thought out by some real leaders in biomechanics. So this whole floor is raised. So I can embed an instrumented treadmill flush to the raised floor. I can put plates, I'll add in the rails, but I can put the plates almost anywhere. So I'm really lucky that the initial footprint of the space has been really well thought out. What else? So we've got the EMG system going in. And the other piece is that this is kind of a bit of a wet space. So some sinks, a sink and some desk space there. So I'm really excited. We're aiming to have this done by the end of this year. The other thing that's really cool and related to the biplane system is that one of the challenges, and that was particularly evident when I was in Calgary, was that the software that you use to reconstruct the kinematics is really time consuming and clunky. So, there's steps when you do the reconstruction. You've got your input images. So what you collect from the biplane system, and then you can have all these different ways of saying the bone is matched or not. And there's all these, it's kind of like a rat's nest of what you can do to tune it. And everybody's own software is kind of home cooked right now. And then eventually you get the joint kinematics. So obviously, if everybody's software is home cooked, it's really difficult to reach that scale up across multi-center studies, which, given how expensive these type of data are to do and how long it takes to reach sample sizes that are going to be big enough to have a real impact in understanding what happens clinically, we need a better way. So we recently got a really competitive score on a collaboration with Mike Rainbow, who's at Queen's University, myself, Trey Crisco, who's at Brown, and we're going to redevelop the Autoscoper software that does all of this tracking. And we're going to do it with Kitware, who's an industry partner. So over the next, whenever we get the funding, the NIH is on a continuing budget, meaning that we're waiting for the government to tell NIH how much money they get so they can tell us how much money we'll get to fund the grant. When we start, it should be about 3 years, and this software will be deployed. It'll be open source. So we're really excited about that. I think those are the kind of top things that are coming down the pipeline. And of course, I want to acknowledge all of my collaborators and all of the funding agencies from when I was in Canada to where I am now. It's been a great journey, and too bad I couldn't do the talk in person, but I'm here and I'm glad to be back in Calgary, and I'm glad that you guys were here to listen. So I will stop sharing, and hopefully there'll be some good questions and discussion from the floor.
Thank you, Dr. Beveridge. That was a great presentation. At this time, I'd kindly ask the Zoom audience members that if you're comfortable to please turn on your Zoom camera to allow for a better discussion period. To ask your question, you can use the raise hand function in Zoom located under reactions. And if you're unable to do that, please type your question in the chat box and I'll read it out loud. I'll actually start off with a question, Jill. I thought your finding, the unexpected finding of changed mobility in the contralateral limb was really interesting. And I was wondering if the onset of post-traumatic OA in these populations would happen in both the reconstructed knee and the contralateral knee typically.
It's a good question. So the answer is no. It typically happens in the operative limb, which again, if both limbs are changing, it really starts to point to the role of injury. So there's something about the clock starts ticking in terms of PTOA at the time of injury. And so I'm actually trying to write a grant, again, using an animal model to try and tease that apart. What is it? What is it about that time that could predict worse cartilage damage in the operative limb or in the injured limb?
Mm-hmm. Awesome, thank you. I'll read a question from the chat box next. Did you look at OA in patients with partial tears as well as complete tears with reconstruction?
So no, I didn't. I know that some of the PhD students and postdocs in Cy Frank's group, from an animal model perspective, looked at that after me and showed that there are similarities in the joint mechanics abnormalities, but that they weren't as severe as complete ACL deficiency. What is not out there is that I am unaware of any animal models that have done reconstructions and looked at joint mechanics. There's some good biplane video radiography data out there on ACL reconstruction kinematics, some of which I showed. And in terms of comparison between different graft types, again, I think there may be a few, I haven't done it, but I think there are some studies out there by Freddie Fu, who's used biplane video radiography to look at different graft types, as well as different ACL procedures, whether it's like single bundle or double bundle. And all of the data out there suggests that if there are any differences, they're very different, or very different. They're very, very small. They're like less than a one millimeter different.
Great, thank you. Art, would you like to ask your question, please?
I believe Smelter was also ahead of me. Wanted to just a little bit. Yes.
You're kind of muffled today, Art.
So I was I've been interested that the result of the idealized graft, and what I was wondering. So I'm thinking like if I'm doing some construction work on my head.
Art, we're having a hard time hearing you today. Maybe you'd want to type your question in the chat box. Awesome. Maybe while Art's typing that, I'll read another question from the chat. For the relationship between the speed displacement between the tibia and femur and the cartilage degradation, do you think one causes the other? Do they both feed into each other in a positive feedback loop? Or do you think there's some causal mechanism in common?
I think that I don't think that there's a positive feedback loop, because one of the things with velocity and sliding velocity, if cartilage is degrading, let's say if cartilage is degrading, you actually get surface roughening, which is actually going to increase the frictional resistance to sliding. And that's also been shown with the decrease in lubricin, which lines the cartilage surface. So I think there's going to be surface roughening and less likelihood, like you'd have to have more force to achieve the same sliding velocity. So I don't think those two things are a feed forward mechanism. Maybe the feed forward mechanism, you're meaning that there's cartilage damage, you have increased sliding velocity, so there's more damage. That relationship, I think, is more causal. Just reading again to make sure I've answered it. So I think that the sliding velocity causes the cartilage damage.
Great. Sorry about that, Art. I'll read your question aloud. Art is interested about idealized ACL graft. He's thinking that maybe things don't come back together the same as original. What happens with idealized graft? And is it possible to put it in such that the mechanical changes are already compensated for?
Thinking about your question, Art. It's certainly possible. I think that ligaments have a certain pretension that as soon as you change that, it's probably nearly impossible to get it just right again. I think that the advantage or the appeal of using the ICLR is that obviously a graft, like a traditional autograft or even allograft, the tension will never be the same. Like you won't even be close. At least with the native one, like you might be closer. So it's kind of like the best case scenario, acknowledging that it will never be exactly the same as if you hadn't mucked with it at all. One thing that we did do was that we did muck with the tension and twist of it. So there was a couple animals, I didn't show the data, and I'm actually not even sure whether it's published, but we actually we pulled that bone core out to add more tension, and we pushed, or in other animals, we pushed the bone core in to create less tension, and then we watched what happened. And so you'd kind of, what happened in the very few animals we tried that from a kinematic standpoint, it's kind of what you would expect. That if you pulled it tighter together, the ranges in degrees of freedom actually were reduced. And if you pushed it in, it was actually closer to pre-injury kinematics. And we saw more damage in the ones that we pulled tight. So it was almost like the tighter grafts were worse than if you were to leave it slightly loose.
Thanks for that answer. Walter, would you like to go ahead with your question, please?
Yes, thank you. I held up my hand there and I wanted to respond. I wanted to respond to Emily's question. So it's a little bit late now, but if you don't mind, I'll come back to that, because I was also intrigued by the fact, that you had the symmetry in the ACLR group, but that group had this more anterior translation. But the movement that you're looking at, if I remember correctly, was within the normal range of motion. It was straight. There was no rotation, no cutting, no nothing. So I assume it has nothing to do with the anterior cruciate ligament, because I don't think it's getting really very much engaged. And so my hunch is that what you're seeing is differences in activation, and that the muscles really control what happens at the knee joint there. And I guess the question is, have you measured EMG to potentially test that particular hypothesis?
Can I share my screen?
Yes, yeah.
Okay.
I'm sure you can. I'm talking for Emily here, but I'm sure she would allow you to share.
So the answer is here, let me just go, because I thought this might come up.
Well, it seems the obvious question.
Okay, so first part. I would actually counter argue that I believe the ACL is in tension and is being challenged in this landing. So the reason is that Lou DeFreight at Duke has shown some really good evidence of what position the joint is in at the time of ACL failure. And he has, when you reconstruct that, the joint is in pure knee extension. And that happens, they've also done some work where they've just done like foot insertion distances. And they're at their maximum interinsertional distance at peak extension just before landing on a one leg hop. So that actually corresponds to the time where I'm detecting these maximal anterior tibial translations. So I would counter argue that I do think the ACL is being challenged at that time point. Now, the other piece, I agree with you that I think the muscle activation is driving this. So what was really interesting, we did EMG on these same subjects. And the only thing from conventional EMG where we looked at time off, time on, co-contraction indices, we really didn't see much other than, this is the ACLR group, we saw a little less hamstring activation, but overall in both left and rights, more hamstring activation compared to control. And then, like I said, we didn't really see much, but then we did some wavelet analysis with Payam Zandia, who was a PhD student with Janet Ronsky. So in these subjects, this is ACLR, this is healthy, we're seeing a dramatic increase in vastus medialis and rectus femoris activation and intensity during takeoff and airborne phases right before landing, which coincides with that timing where I'm seeing anterior tibial displacement. And the other piece to this, I'm showing you just the ACL reconstructed limb. We've now done so actually this paper just got accepted in Journal of Biomechanics, so it should be coming out any day. But what's interesting is that we looked at the contralateral is doing the same thing. And so
one thing, of course, that you have to be careful when you say that peak EMG occurred exactly when I saw that peak rotation, anterior translation. You know, there's a force delay there, an electromechanical delay.
That's fair.
Yeah.
That's fair.
But I would agree with you that what happens before the landing determines what happens right at the landing, because, you know, we activate our muscles in walking, in running, in jumping, in landing before we actually need the force, because if you only activated them once we hit the ground, we will collapse because the force increase is so slow. I have many other questions, but I want to have other people have a chance as well. But one question that I really want to ask is, I was really surprised that your vertical ground reaction forces in the animals were much greater 4 weeks after the surgery compared to 20 weeks when they were lower, because in all the animal models that I've worked with, I see the opposite. I see smaller ground reaction forces right after surgery, and then the ground reaction force recovers. Do you have an explanation for that?
Nope.
No?
I don't.
Okay. Well, it's interesting and definitely, unusual.
Yeah. So what we're doing right now is we are using a tech scan pressure walkway, which if you use just their built-in software, it gives you like these really discrete variables that are really not helpful. And so we actually took that output and kind of wrote our own software so we could get those force curves. And for the same reason you just brought up, Walter, we're like, well, I'm not sure we know what's going on. It'd be great to get more data. So we've asked some collaborators at Brown who are using a very similar drill model to share their tech scan data with us so that we could actually increase our N to get a better sense of what is going on. Because we only actually had 3. This was a really preliminary study to figure out how do we make all these nuts and bolts go with the staff and the resources that we had at Cleveland. So we're just actually trying to figure out the answers to your questions.
Great, yeah. Thanks a lot. Thanks for the talk. Enjoyed it a lot.
Great.
Brent, please go ahead.
I have just one quick one and then a bit of a longer one. But my quick question is, when you do the analysis to look at the ACL graft stiffness, are you also able to look at the relative velocity between the 2 joints and to see if there's a relationship between the stiffness of the graft and that relative velocity measurement?
That's what I'm trying to get to, because I think that would be informative. And so trying to put all of that together is again what I'm working towards. With the biplane video radiography data in the clinical subjects, one of the dirty little secrets about those data are that there's actually quite a bit of noise in it from the bone matching. And so it's almost like it jitters. That's my best sense to describe it. And so extracting velocities is, I don't have a lot of confidence. And so that's actually why you don't see a lot of it published in those types of data. And also why we're hoping with this software development, we can make it a little bit better. So with the animal models, I haven't been able to do it all together all at once, but that's what I'm trying to get to.
Yeah, okay, all right. So my other question is, I don't study osteoarthritis. It's too complex of a disease for me. But one of the things that's pretty impressive to me, especially when you talk about knee osteoarthritis, is all of the studies that have seemed to characterize that CAM measurement, the knee adduction moment or whatever, it's pretty consistent with the ability of that surrogate measure of joint loading to predict progression of osteoarthritis, who needs a joint replacement later on. And I even saw a recent paper that sort of was able to predict people that went on to get osteoarthritis from a pre-arthritic state. So there's something to this CAM measurement, and I think it has something to do with overloading of the knee joint leading to OA. But then my question is, so like yesterday, Dave Saxby's group out of Griffith University published a paper in osteoarthritis and cartilage. And they did a clinical trial where they looked at people with hip osteoarthritis, and they got them to walk with larger stride lengths, and it increased their joint loading at the hip. And their pain went down. It was like N equals 20. And so they're trying to say that if you have hip OA, you want to increase loading, and that's good. And if you have knee OA, you have to decrease joint loading, and that's good. So I don't know what the question is here. I guess my question for you is, do you think that there's different mechanisms for why somebody would get OA at the knee versus OA at the hip? And I guess before you answer that, I'll just tell you that my comment's going to be that if so, I don't like that. And if that is the case, I think we need to change the name from osteoarthritis to, we need to be calling these two different things. But where do you stand within the argument of overloading, underloading, cartilage, OA progression? Maybe that's more my question. Thank you.
Well, that's a good one. Let me I mean, I think this is a discussion more than a question.
Yeah.
For all of us, but I think that one of the challenges is that what kind of scale you look at, what kind of measurement you do, when you do it after injury, where you do it in that trajectory. Like, are you at end stage, or are you at like before all of this fall, like the wheels start to fall off? I'm going to wager that you're going to get different answers depending on what you use to look at, when you do it, and which joint. So, and also what the injury is. For my thought is that there's groups in Delaware that have used these like EMG driven models and inverse dynamics to say like, yes, unloading after ACL injury is bad. And so you could argue that, well, we also know cartilage, degenerates if you are on bed rest. So like there is an element of unloading that is bad. But if you go down one scale further in the unload versus offload, my thought is that if you're doing inverse dynamics, you may be offloading the joint from that scale. But if you drill down to the contact mechanics, you may actually have an alignment and incongruity where you have this really small increased loading that is actually damaging the cartilage at a really small point. And all it takes is that one small area. So as a whole, you may be unloading it, but when you look at like a really smaller scale, you may be overloading it. So I think that may be important. So maybe early underloading may be bad, but you may be doing it for a reason that we don't totally understand. And then by the time you have an increase in adduction, like, the CAM measure, which I agree is a good predictor of rapid joint degeneration, is that like, has the tissue properties already started to change? Have the meniscus compressive properties already changed that allow increased displacement out of the joint? Has the cartilage already started undergoing more compression? So as your CAM increases, the wheels have already started to fall off. So it's actually just following the changes in joint tissue properties that get worse and worse and worse. So I think that, and again, that may be downstream of what's happening at the very initiation. And I think that trying to link and create a continuum, this is where creating continuum from the animal model to the clinical model and trying to use similar tools across, it's really hard to do. And it's really expensive. But I think it's really important, because then we can start to tie these disparate results together that form a more cohesive story. So that's where I've really kind of tried to, that's my vision, is to be able to do that with my research program.
Yeah, I guess one of the problems that I have with like fixing this disease or curing this disease is we can't even decide what causes it or understand what causes it at the ankle versus the knee versus the hip in a human. And then that makes me then question every single animal model out there in the ability to translate the findings from any animal model to the human condition, just because it's like, which human condition? The hip human condition, the ankle human condition,
and that's where I think
the PTOA human condition.
We could go on forever. But I have some points about that, and choosing the right animal model and making sure that you are aware of what you're picking as your animal model, I think is really important. And being very thoughtful in that, and not picking it for ease. So like I said, Brent, you and I think we could, ORS is coming up. So we'll have a drink and then we can continue our debate.
All right, that sounds good. Thank you.
Anytime.
We're just about out of time, but maybe our last question, I'll read one from the chat box from Kelsey Collins. Are you thinking about changes in mechanical properties of other joint tissues? Intrapatellar fat pad, synovium thickening, inflammation that could happen with injury or OA progression or after surgery? Could this be affecting the loading behavior of the joint or graft?
So yes, although I didn't touch on it, all of these, again, trying to leverage the best of both worlds, is that we do collect synovium, we do collect fat pad, we do collect these tissues. And so we are poised to be able to ask some of these questions. And this, at the start of the talk, I kind of had a dashed box around biology because I'm starting to wade into it, realizing that because I'm trying to look really upstream of this disease pathogenesis, there's a lot of evidence out there that those tissues play a really important role in setting the stage for whatever happens to the mechanics. Because if you have an inflamed tissue that already has impaired ability to bear load because of the inflammation, even normal loading will cause damage. So I think that all of those other tissues and thinking about the joint as an organ is really critical. And it is complex, and the bone's going to talk to the cartilage, and the synovium is going to talk to all of the intraarticular tissues. So yeah, my short answer is yes.
Just to follow up, I don't want to butt in, and I'm not going to take Brent's bait, even though I'm tempted to, but I think that this is so cool because there's so much hand waving about mechanobiology, and it either is largely measuring biology and inferring mechanics or the converse. And I think that this setup that you have with the collaborations that you have, I think you're really well positioned to actually measure mechanobiology in a really pure way that I think is really clinically translatable. And for that reason, I think it's super exciting. So everyone knows I'm the president of the Jill Beveridge fan club, but I think this work is really great.
Well, so since Kelsey's butting in, I'm going to butt in as well. And my contribution would be that, we measured mechanical properties of articular cartilage, 4 weeks, 8 weeks, 10 weeks. And so after ACL transection in rabbit and cat models. And what you see there is that there's a distinct swelling of the cartilage within a very short time, within 4 weeks, and a loss of stiffness by about 50%. So the stiffness is about half after 4 weeks. And therefore, when we then applied exactly the same force through the joint in exactly the same joint configuration, the force distribution across the articular cartilage was completely different. The peak pressures were dramatically reduced and the contact areas were much greater than before the anticruciate ligament transection. I think this is what Kelsey raised there, was one of my points as well. I think it's very important to get a handle on that, because the same loading from the muscles and vertical ground reaction forces and so might not ensure at all, and in fact probably doesn't after ACL and after ACL reconstruction, that you have the same joint pressure distribution in the joint, because your cartilage dramatically changes. It changes enormously dramatically and measurable day by day when you unload. And then when you load, it changes very, 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 very slowly. And you have to wait years before you lose all the articular cartilage. So I'm definitely a fan of unloading is really, really bad.
Yeah, I completely agree. And actually I'm trying to write that concept of like, if the inflammation is happening really early and changing the mechanical properties from the outset, no matter what you do mechanically, the joint may be doomed. So addressing the inflammation early on, and how do you do that? And some of to the point of getting the right animal model is there's been a lot of work and interest in these intra-articular injections of anti-inflammatories given straight after injury to try and prevent this degradative process. But the animal models that we've been using are like DMM, complete ACL transection. Like no matter what you put in the joint, once it's inflamed, if the mechanics are that terrible and that's your animal model, of course it's going to fail. Like, of course it's going to be bad. We don't leave people without a functioning medial meniscus, like a damaged medial meniscus root horn. So I think that I totally agree that these tissues, the mechanical properties of the tissues changing really early on we have to address it, and we have to have the right tools to be able to do that in animal models, and then to be able to translate that. So that's an area that I'm really interested in, and figuring out how to do that well. I actually just added some of your references, Walter, into my into my grant.
Well, as you know, if Kelsey is the president of the fan club, or Jill, then I'm the vice president.
Well, I love that I'm being loved in Calgary, because I still cherish my time here, and you guys have such a great program. And so for all the trainees listening, like, you guys are really fortunate to be where you are.
Great. If there are no final questions, we'll end there. Dr. Beveridge, thank you so much for your presentation, and thank you to all the audience members for the great question and answer period. I hope to see everyone again next week for the HPL seminar, and we'll be hearing from Dr. Kelsey Lucas. So I hope to see you then, and I hope everyone has a great week.